Picture this, it's the 1600s in Japan. You're a political exile banished to Hachijojima, a remote, incredibly harsh coastal island. You're facing severe malnutrition, forced labor, but against all odds, you survive. Not only that, you actually maintain this extraordinary physical stamina. How is that possible? Well, historical records point to a resilient local plant they used as a daily survival food. It's called Angelica kiske, but you probably know it as Ashitaba, or tomorrow's leaf. It gets that name because, literally, if you pluck a leaf today, a brand new shoot visibly sprouts by tomorrow. Pretty wild, right? But the question is, does this botanical folklore actually hold up under the microscope of modern science? Welcome to our explainer. Today, we're cutting through all the commercial hype to bring you a truly rigorous, academically oriented look at Ashitaba's true pharmacology, its clinical efficacy, and its geroprotective mechanisms. Okay, let's dive right into this syllabus. We're going to systematically move from the molecule up to the human clinical realities. We'll cover ethnobotany and phytochemistry, geroprotective mechanisms, human efficacy, cardiovascular and oncological impacts, toxicology and safety, and finally, the longevity hierarchy. Section 1. Ethnobotany and phytochemical profiling, focusing on the yellow sap and chalcones. Now, one absolutely crucial detail you need to understand right out of the gate is that commercial Ashitaba isn't just one uniform thing. Those generic, fiber-rich leaf powders you see, they completely pale in comparison to the highly potent, thick yellow sap that aggressively oozes from the plant stems when you cut them. Let's move to the timeline and see how our understanding builds here. We kick off way back in the 1600s with those Edo period exiles surviving on the plant. Fast forward centuries later to 2007, where we get the very first human trial looking at Ashitaba and blood glucose. Jump to 2019, and boom, a landmark paper in Nature Communications discovers a specific compound's ability to trigger autophagy. And that brings us to the present, with a 2024 randomized controlled trial looking at visceral fat reduction in humans. So what's driving all of this? It's a unique class of molecules called chalcones. These are open-chain flavonoid polyphenols that are heavily concentrated in that yellow stem sap. The big three heavy hitters we're tracking today are 4-HD, XAG, and DMC. Now, what makes these specific chalcones so biologically powerful is their highly lipophilic prenylated structures. In plain English, their chemical shape is basically a VIP pass. It allows them to seamlessly slip into and cross our cells' lipid membranes, which dramatically enhances their systemic bioavailability compared to your standard dietary flavonoids. But wait, before we get into the really cool anti-aging science, we've got to address a very highly debated question. Is Ashitaba a reliable botanical source of vitamin B12? You've probably seen this pervasive claim plastered all over vegan supplements, pitching the plant as this rare, holy grail terrestrial source of B12. Well, the rigorous analytical data paints a completely different picture. While true, bioactive cobalamin is absolutely essential for your DNA synthesis and keeping your nerves healthy, what we actually detect in botanicals like Ashitaba is something called pseudovitamin B12. Basically, it tricks outdated microbiological tests into reading positive. It's literally like handing your cellular metabolism a fake ID. The bouncer is going to completely reject it. And it gets worse. Not only is it biologically useless to humans, it actually competitively binds to your transport proteins, meaning it actively blocks the absorption of any real active B12 you might consume. So no, standard leaf powder is definitely not a reliable monotherapy for B12 deficiency. Moving right along to Section 2, mechanisms of geroprotection, we're talking cellular fasting and AMPK. Now, this preclinical biology is where things get incredibly exciting. To understand how these molecules alter aging, we first have to understand the main goal here, and that is autophagy. Think of this as the cell's premier intracellular recycling and quality control system. As we age, our cells get cluttered with toxic garbage, like misfolded proteins and damaged organelles. Autophagy is the janitor that rounds up all that cellular trash, degrades it, and magically recycles it into fresh bioenergetic building blocks. The problem is, this whole recycling process naturally slows down as we get older. And that's exactly where the chalcone DMC enters the picture. Research from 2019 demonstrated that DMC acts as a potent cellular fasting mimic. Here's how it works. You ingest DMC, and it modulates specific GOBTA transcription factors. This essentially tricks the cell into going into a defensive, self-renewing survival state of nutrient scarcity, which then induces massive systemic autophagy. And the absolute coolest part? It pulls this off entirely independent of the usual mTRC1 pathway, which is how it managed to extend lifespan in yeast worms and flies in those lab models. And this brilliantly illustrates the other side of the metabolic coin. While DMC triggers the recycling, the other two chalcones we mentioned, 4-HD and XAG, go to work on lipid metabolism. They directly phosphorylate AMPK, which is basically the master energy sensor of your cells. By activating AMPK, these molecules forcefully shift your cellular metabolism away from creating new fat, a process called lipogenesis, and push it toward burning existing fat stores through mitochondrial fatty acid beta oxidation. Basically, they suppress adipocyte differentiation, making them extremely potent anti-adiposogenic agents. All right, Section 3, Human Efficacy and Metabolism. It is time for a controlled narrative pivot to the clinical reality. Now, what's really interesting about this slide is that number right there, 12 weeks. We have to step away from the enthusiasm of the animal models and ground ourselves in human data. 12 weeks is the entire duration of the strongest positive human trial we currently have. In 2024, a randomized, double-blind, placebo-controlled trial found that overweight adults taking encapsulated chalcones saw a significant reduction in their visceral fat area. But let's be real, 12 weeks is a very, very short window when we're talking about the grand scheme of human longevity. Let's impartially walk through the broader clinical landscape. So that 2024 trial showed reduced visceral fat, right? But remember, that's a surrogate anti-obesity endpoint, not a direct longevity marker. Then we have a 2018 study on metabolic syndrome that showed lowered ghrelin, the hunger hormone, and some fat reduction, but that was only seen in males, and it was a largely exploratory study. Before that, a 2007 trial suggested lowered glucose curves, but that evidence literally only exists at the abstract level. So just to firmly temper our expectations here, human efficacy is strictly limited to modest metabolic and body composition endpoints. There is absolutely zero human data proving it extends our lifespan. Section 4, Cardiovascular and Oncological Impacts. Let's look beyond metabolism. This next part is critical because patients with metabolic syndrome are highly vulnerable to thrombotic diseases. The findings here are fascinating. Ashitaba chalcones, specifically XAG and 4-HD, actively preserve the blood's native anticoagulant status. How? They do it by inhibiting the synthesis of thromboxane B2, which drops the rate of pathological platelet aggregation. At the exact same time, they suppress the inflammatory overproduction of a protein called PAI1. Usually, PAI1 represses your body's natural clot-dissolving system. So in metabolically impaired models, Ashitaba acts almost like a traffic cop, helping to keep your blood flowing smoothly. And the preclinical data gets even more sophisticated when we look at oncology, specifically showing dual-targeted action against aggressive malignancies like melanoma in mice. Let's break it down. Step 1. 4-HD and XAG simultaneously bind and suppress two hyperactive mutant kinases that drive tumor growth, BRAF and PI3K. Step 2, this double blockade induces a G1 phase cell cycle arrest, effectively hitting the brakes on tumor division. And step 3, it triggers intrinsic apoptosis, which is programmed cell death inside the tumor cells. It's an incredibly elegant, multi-target mechanism that standard single-target therapies honestly struggle to achieve. Moving on to Section 5, Toxicology and Safety. These are the critical parameters we have to respect. Look, precisely because these are biologically powerful molecules, they require strict clinical caution. Here are the contraindications you need to know. The absolute major risk here is combining Ashitaba with oral anticoagulants, particularly warfarin. Ashitaba can inhibit the CYP2C9 enzyme, which is the exact enzyme responsible for metabolizing warfarin. This completely amplifies the drug's effect. Add that to Ashitaba's own antiplatelet action we just covered, and you create a highly dangerous synergistic bleeding risk. Also, if taken with diabetes meds, you could see additive drops in blood sugar. High doses might even trigger phototoxicity, giving you severe sunburns due to plant compounds called furanocoumarins. And finally, because we just don't have the safety data, it is explicitly restricted during pregnancy and lactation. To put a number on that safety profile, the European Food Safety Authority thoroughly evaluated concentrated Ashitaba sap powder. Operating under strict precautionary principles, they authorized it as a novel food supplement for adults, but they set a highly conservative maximum intake level of just 137 milligrams per day of the pure sap powder. Finally, Section 6, the longevity hierarchy. So the crucial point is, what is the bottom line here? After going through the ethnobotany, that thrilling preclinical data, and the modest human trials, where does Ashitaba actually fit in your toolkit? Let's compare it directly to established interventions. Exercise? High evidence for human longevity, vastly outperforming Ashitaba. Calorie restriction? Moderate to high mechanistic and human data. Pharmaceuticals like rapamycin and metformin have moderate human evidence but are far better developed geroscience candidates. Ashitaba currently sits near the very bottom of the hierarchy for actual longevity evidence. It's an undeniably fascinating botanical, but right now it is best framed as a speculative metabolic adjunct, not a frontline anti-aging intervention. Which leaves us pondering this final, vital question. Does the rush to commercialize complex botanicals outpace our rigorous understanding of human aging biology? Ashitaba is a remarkably resilient plant, and its potential for metabolic health and cellular biology is legitimately incredible. But true longevity science takes time. It takes massive clinical trials and immense patience. Keep your expectations grounded, stay curious, and always track the translational research, not just the marketing. Thank you for joining me on this explainer.